So describe the structure and major function of phospholipids and the plasma membrane. So we're going to talk about it here as it pertains to membrane transport, but this is actually a topic that's found in chapter 2 in your book. So be on the lookout for that. So we're going to start with a basic review of the cell membrane structure, and then we're going to move into the actual chapter 4, which is membrane transport. Also, I've shared a video on the Brightspace page that kind of breaks down and simplifies cellular respiration. So if you're having difficulty with any of the processes, glycolysis, oxidative phosphorylation, or understanding the difference between substrate level phosphorylation and oxidative phosphorylation, I would highly recommend you look at that video. It's also linked in the PowerPoint slides in that packet, in the cellular metabolism packet as well. So have a look at that video. It may help to clear things up, simplify it a little bit if you're having difficulty with that material. So let's move on here. So we're going to start with that first objective, which is basically talking about the structure and function of the plasma membrane. So this is pretty much a review, just kind of reminding ourselves of what the cell looks like, what the cell membrane is made of, what some of the organelles inside the cell are, their functioning, as it pertains to understanding membrane transport. So in order to understand how molecules move across the plasma membrane, we really want to start by reviewing what the plasma membrane looks like, what's going on in the plasma membrane. So the description of the plasma membrane is called the fluid mosaic model. The fluid mosaic model. And this is basically a description of what the structures in the plasma membrane are and how they are fluid and flexible, and how they change in order to facilitate cell functioning, specifically cell transport, cell membrane transport. So we call this a fluid mosaic model because it's not a fixed structure. It's not a constant structure. This plasma membrane is continually changing itself. We're moving new proteins in, we're moving old proteins out, we're changing the amount of cholesterol in the membrane. So it's a fluid situation happening here. It's very flexible to what the needs of the cell are and to what the environment in which the cell is as well. And so that's why we use the term fluid mosaic model. We know that the main structures of the fluid mosaic model are these fat molecules that we talked about already. We call them phospholipids. And so the structure of the cell membrane is really this bilayer of phospholipids. We're going to draw that out here. And we've got these amphipathic phospholipids, which have the polar heads that face the aqueous environment and the non-polar tails that face the internal layer of that bilayer, or the internal surface of that bilayer. And so we've got this double layer of phospholipids here. Okay, now studied along the phospholipids, we also have some other very important structures. First off, we have proteins. So we have some proteins that go right through the membrane, and we'll describe these here in a little bit. We're going to talk about what we call these, but some of them go right through the entire span of this bilayer. We've got some proteins that are embedded just in one side of the bilayer. Oops. We've got some that are on the polar end, so facing the internal environment of the cell. Let's actually label this out. So this is going to be our intracellular environment, our extracellular fluid compartment out here, intracellular fluid compartment down here. So inside the cell and then this is outside the cell. We've also got some ringed cholesterol structures. We'll talk about the function of these in the bilayer here in a bit. But if you remember the structure of cholesterol, which are these ring-like structures. Okay. And then we also have some proteins that attach carbohydrates. In carbohydrates, we talked about these glycogen branching, these polysaccharide units. And we can also see this branching happening on the phospholipids themselves as well. And we'll talk about the function of these. So this is kind of a little bit what the plasma membrane looks like. So let's start out by describing each of these structures and talk about what their importance is in the plasma membrane. So first off, we have the actual phospholipid bilayer. And just to remind ourselves, this is the polar head of the phospholipid, and this is the non-polar tail. Okay. So the phospholipid bilayer, a couple of features here is that it's the main structure. So the main structures in the plasma membrane are the actual phospholipids. They are also amphipathic, so we talked about what that means, but this is what allows them to be the perfect structure for this function. They can face the aqueous environment on one end and the non-aqueous environment on the other end. The third thing is that they offer fluidity and flexibility to the membrane. So because they're not rigid structures, these partly polar, partly non-polar, they offer fluidity and flexibility to the membrane. And then we also know about them, they are impermeable to polar structures, but they are permeable to non-polar structures. So we talked about the fact that if a molecule is not, is polar, it cannot exist in the center of this bilayer. So a polar molecule cannot exist in the non-polar hydrophobic region within here. So therefore it cannot traverse or pass or go through the plasma membrane. Only non-polar items or non-polar molecules can traverse the plasma membrane. So it's impermeable to water and other polar, molecules. Impermeable to water and polar molecules. Okay, so that's the first structure, the main structure of our plasma membrane. Let's talk about some of our proteins. So we have proteins that go right through the plasma membrane, which we drew here. This is called an integral protein. And an integral protein means a couple of things. So let's draw this out or describe this here. So an integral protein is just what that word means. It is integral to the structure of the plasma membrane. It is a permanently fixed structure. So the plasma membrane cannot exist without this protein. So it's permanent, it's a permanent permanently fixed structure in the membrane. It's a transmembrane protein as well, meaning that it spans both sides of Okay, so our integral proteins are crucial to the structure of the plasma membrane. The cell will die if this protein is moved from the bilayer. And so this is integral to the function and structure of the protein. We can compare that to peripheral proteins. So this is an example of a peripheral protein. And the peripheral protein exists on the cytosolic surface or the cytosolic side. So this protein exists on the surface within the cell, inside the cell. And this is not a permanently fixed protein. This can be removed, replaced. It can easily dissociate and move around the plasma membrane and move from one spot to the other. So this can easily dissociate. And move around. Okay. So those are our two types of proteins. We've got integral proteins that are crucial to the structure and function of the membrane, and then peripheral proteins that are temporary proteins. They're on the cytosolic side within the cell, and then they can move around easily. Let's talk about cholesterol and the function of cholesterol in the membrane. So cholesterol was one of our fats that we talked about. Cholesterol is also going to increase the fluidity and flexibility of the membrane. And the reason is cholesterol comes in and breaks up the phospholipid interactions, and it prevents the membrane from becoming this rigid, hard structure by breaking up, oops. Okay. So it breaks up the phospholipid interactions, and so it decreases the rigidity or the crystallization of the membrane by allowing the membrane to have some fluidity from these cholesterol. Hang on here. Okay. Some fluidity from these cholesterol structures. So it's going to increase fluidity and flexibility, which is basically decreasing the rigidity or crystallization. Okay. It also prevents the membrane from leaking, so it kind of acts as a safety net, if you will, and prevents structures from leaking across the membrane. Okay. It is also impermeable to polar molecules and to water. Remember, it's a fat, so it's going to act just like the hydrophobic center or the hydrophobic tails in that it prevents the passage of water and polar molecules. Okay. Hence, it's going to prevent leakage across the membrane. It's going to maintain the membrane integrity. The final structures we're going to talk about are carbohydrates. Let's try to put it up here. So carbohydrates are also structures within the cell membrane. We have two types. They're basically covalent linking of these branched carbohydrate chains, these polysaccharide chains, either to the phospholipid, and we call them glycolipids, or to the proteins in the membrane, and we call them glycoproteins, okay? So here's a glycoprotein. If we have these carbs linked to the proteins in the membrane, and here's a glycolipid if we have these carbs linked to the phospholipids in the membrane. And so these carbohydrates, we said, are covalent bonding to proteins or phospholipids. And these help with things like cell tagging or cell identification. They help one cell to signal to the other. So basically cell tagging and cell communication. These are what these are the functions of the glycoproteins and glycolipids. So cell tagging and cell communication. Okay. So this is the structure of our cell membrane. These are the various types of proteins that we can have. Okay. We can have integral proteins, peripheral proteins. We've got cholesterol. We've got proteins. We've got, carbohydrates that are bound to either proteins or lipids. And then we've got the main star of the show, which are the amphipathic phospholipids. Okay. The important thing I really want you to remember here is the reason that polar substances cannot go through the membrane, because they cannot exist within the hydrophobic center where we have these non-polar tails, the fatty acid tails of these phospholipids, okay? Really, really important part of understanding the function and how things can move in and out of the cell through this structure,